So hello everyone, welcome to dkNET webinar. I'm Koei, I'm the host of today's webinar. For those of you who are new to dkNET, dkNET is a research resource information portal funded by NIDDK, and dkNET have several services for you to discover, including supporting rigor reproducibility. We also help research to discover useful research resources, and then we have Hypothesis Center for you to generate hypotheses, help you doing that. And also we have fair data management service to help research to make your data fair. So today we are very excited to launch our new webinar series featuring fair data management and useful resources. Before we start the webinar, there are a few housekeeping items to cover. Please note that this webinar is being recorded. The webinar recording will be shared on dkNET YouTube channel next week. Please mute yourself during webinar presentation. If you have any question during presentation, please use the chat function. After presentation, you can unmute yourself and ask questions during your Q&A session. So in today's webinar, it's our pleasure to have Dr. Susan Gregurick join us and give us a presentation on creating and sustaining a fair biomedical data ecosystem. Dr. Gregurick was appointed Associate Director for Data Science and Director of the Office of Data Science Strategy, OTSS, at National Institute of Health on September 16, 2019. Under Dr. Gregurick's leadership, the ODSS leads the implementation of the NIH Strategic Plan for Data Science through scientific, technical, and operational collaboration with the institutes, centers, and offices that comprise NIH. Dr. Gregurick was instrumental in the creation of the ODSS in 2018 and served as a senior advisor to the office until being named to her current position. Dr. Gregurick was previously the Division Director for Biophysics, Biomedical Technology, and Computational Biosciences at the National Institute of General Medical Sciences. Prior to joining the NIH in 2013, Dr. Gregurick was a Program Director in the Office of Biological and Environmental Research at the Department of Energy. Before beginning a career of government service, Dr. Gregurick was a professor of computational chemistry at the University of Maryland, Baltimore County. Her research interests including dynamics of large biological macromolecules, and her areas of expertise are computational biology, high-performance computing, neutron scattering, and bioinformatics. Dr. Gregurick received her undergraduate degree in chemistry and mathematics from the University of Michigan, and her Ph.D. in physical chemistry from the University of Maryland. So without any further ado, let's welcome our speaker today, Dr. Susan Gregurick. Susan, over to you.
Thank you so much, Koei, and to all of my friends at dkNET. Thank you so much for the work that you do to support fair data management, data sharing, data repositories. These are really key activities, and we really appreciate the work that you're doing. I'm really pleased to tell you what we've been doing this past year to create and to sustain a fair biomedical data ecosystem. This isn't work by one person, it's work by many, many people that span all of our institutes and centers at NIH. And so I'm just representing the work of at least 80 people, and it's a real pleasure to lead and to coordinate NIH in our strategic plan for data science. I don't need to tell you about what FAIR is, your leaders in FAIR. This is just to level set because we're going to talk a little bit about what we're doing to make data, to make resources, to make repositories, to make software, and to enable a next generation of computational and informatics researchers truly cognizant of FAIR. You know, and it seems like if we get there, yes, this is it, we've won. We have made our data, our software, our methods, our models findable, accessible, interoperable, and reusable. So that seems like that would be the goal. Or is it more like this? I pose it to you, and I think maybe you'll agree that really that's just the starting point of a whole bunch of really fantastic science that we can enable because we can find and use and reuse and make data truly interoperable. And this is what I'm really going to talk about, is what we're doing to get to this point, and maybe just a little bit beyond. And if I have time, I will tell you how we're doing this work to address COVID, because I think that there are most likely all of us sitting in our homes or alone in our offices, and we want to get to a different place. You know, NIH supports so many different types of biomedical research. I'm showing you snapshots from many different areas, from structural biology, my field of structural biology, looking at high resolution or low resolution of protein structures. There's neuroimaging, there's pathways and knowledge and circuitry to develop with these data and from the brain. There's interesting and diverse data sets with respect to cell biology, tissue reengineering. And then there's the world of the clinical studies of bio and behavioral studies, and all of these different types of research communities have different types of data sets and very different types of expectations when it comes to making their data fair. And yet we have to level set all of these different communities. You know, sometimes it feels a little overwhelming. There's so much data, sort of like the rhyme of the ancient mariner. How will we come to grips with this? How will we enable a fair data biological ecosystem? And what will we do with that data? There's a proliferation of data, but there's also another rise in the scientific community, which is that of computational resources and algorithms. The merging and convergence of computational work with cloud computing and new types of processors, such as GPUs, and the rise of the Internet of Things with fog and edge computing, that really does converge on data science and computational science. And there's a lot of room for discovery in these new areas. So some people know that I'm a bit of a closet librarian. I used to love to go to the stacks at the University of Michigan, where I got my undergraduate degree, and read all the different new articles that came out during the week. And this is just an example that I'm showing you from a particular study of the retinal ganglial cells. And you can see that this publication has a number of wonderful high resolution images associated with it. And with that, those high resolution images comes data. This is taken exactly from a pilot that I will talk about in a minute from the Figshare Generalist Repository platform that we ran over the past year. And we linked those data sets in Figshare with the publication. This is something that's done, I think, fairly routinely, but wouldn't it be also wonderful if with this type of linking from the repository to the journal article, there was also a way to find and access that data via computable metadata? I think this is definitely within the very soon horizon. And wouldn't it be wonderful if from any of those repositories, I could link that data to any of the algorithms or tools that were used to generate the data, analyze the data, or to make that data reusable. And any of the platforms that I'm interested in using, be they Jupyter Notebooks, Galaxy, or Bioconductor, or even just GitHub. So creating a way to think about journals, repositories, computational platforms in an integrated and more holistic manner is a way to really live fair data. Through myself and a number of my colleagues, we've done a listening tour to all of our NIH institutes and centers, and I'm just going to tell you a little bit about what we learned about NIDDK. You know NIDDK far better than I do, supporting the research on diabetes, endocrinology, metabolic diseases, digestive diseases. NIDDK researchers study a number of diseases and have a number of very impactful platforms, including dkNET. And here is just some of the use cases that they told us they would like to be able to do with data from or with their studies. This is the first use case. It's from a renal researcher who would like to look at the gene expression data coming from various parts of the kidney and mapping that with the data that's in the GTEx database, where this is gene expression data with specific genomic variants. So these two data repositories are completely different, it would be pretty much a labor of love if you wanted to try to link the expression data from the kidneys with the GTEx data set. This is something that I think we can do, and I'm going to show you a little bit about how we might be able to enable a use case such as this. The second use case comes from a researcher who's studying diabetes. And here, instead of looking at a cohort population and studying kidney, they want to do something that's very specific to one individual, but is multimodal data over time. And what this researcher would like to do is to take the same subject, their data samples, in this case, it's data related to diabetes, over many time steps. And this is something that I've heard quite a bit. So it's genomics, epigenomics, transcriptomics, proteomics, metabolomics of one individual over many time steps. Now, the problem is that in some cases, each time a data set is released, it's released with a different subject identifier. So this is the case of linking participants either within one repository across time or participants that have data distributed in multiple repositories. And now I'm going to show you at the very end how we're looking to pilot addressing this issue. So these are just some of the use cases that researchers would really like to get to. And here's how we hope to address these. And addressing use cases means that we'll have more use cases. So it's a wonderful iterative loop. The first thing I'm going to tell you is this is probably going to be the most important component of my talk. So I'll just tell you right up front. And it's exemplified by this figure, which just basically tells you what you probably already know, which is looking over the course of maybe 10 years, NIH researchers who are putting publications into PubMed Central and linking with those publications a data availability statement. It could be any type of statement. I have my data on an FTP server. I put my data in PDB or in Geo. So we noticed that researchers are embracing the concept of making data fair. And as a response, we will fairly soon issue an NIH data management and sharing policy. You've hopefully seen and responded to a number of requests for information about draft policies. The general statement about the policy is that researchers who are NIH funded via R01, a center, a postdoctoral award, or even if you're an intramural NIH researcher, your research will need to have a data management and sharing plan that will be submitted at the time of your application. Now, it may not be scored, but it may be reviewed by your review committee. These plans should explain how you as a scientist will share and manage the data that is a result of your work. Not all data needs to be shared. Certainly that's going to be the case, but the data that you do intend to share, you need to tell us how you're going to share that. The good news is that you will be able to submit a budget that will help you enable fair data sharing. And I think the work that dkNET is doing to help researchers develop those data management sharing plans is critical. It's really absolutely needed, and that will be coming out in some of the pilot studies that we've done. So that was the most important thing I was going to say today, and everything else now is just how do we enable researchers to get to that point, and what can we do if we get to that point? And the first thing to note is that there are many different ways for researchers to share data. If you want, you can attach your data as supplemental materials up to 2 gigabytes with your article, and it will be stored at PubMed Central. There are a number of commercial and nonprofit repositories that will share data and offer many wonderful services. We have a list of all of those nonprofit and commercial repositories for you to generally share data. There is the possibility of data sharing through our STRIDES partnership. I'll explain that in a minute. It comes with a lot of caveats. But as a first stance, NIH strongly, in the strongest possible way, encourages you to please use your community supported data sharing repositories, because these are the repositories that your community and you support share. They have metadata standards, and they support the community in many ways. And here's just a list of a few of them. I hope that some of them stand out. We are supporting many different repositories. And if you ever need to find a repository, you know that you can go to dkNET, organized by repository type and scientific area. And thank you again for the work that you are doing. It's wonderful. There's also a list on the BMIC website for all of the NIH supported open access data sharing repositories, as well as the generalist repositories. And then fair sharing, of course, fair sharing of fair data is another opportunity for you to find different repositories. We, NIH, recognize that repositories and knowledge bases are the bedrock of data science, and we need to support these. We recognize that data resources are important tools. Historically, there's been a wild west of mechanisms at NIH to support these repositories, leading to many different types of issues with respect to management and review. So our work here in the past years to optimize the funding mechanism so that researchers will have a confidence in the data and the information integrity. And so I'm going to tell you about the FOAs that are on the street now to support data repositories and knowledge bases. And there are 4 elements to those FOAs: scientific impact within and across communities, community engagement with your community and with your journals, the quality of the data science and the efficiency of operations, and then a governance. These are the 2 funding opportunity announcements. I am very pleased to tell you that we received our first cohort of applicants just this past week. We have had a strong response from the institutes and centers that are supporting these FOAs. The applications will be reviewed in March, and we'll be able to report outcomes in the springtime. And the next receipt date, I believe, is in January. So do please let your communities know. I will have to caveat that not every institute and center at NIH has signed on to this FOA. We hope that we will be able to have additional institutes and centers signing on as the value of this mechanism and this opportunity is realized. I mentioned that we had a pilot with the generalist repository Figshare, and we did this because NIH, we wanted to understand in what ways would researchers, both in the external community and in the internal community at NIH, would use a repository. What were the gaps and where were our opportunities? And so this is some of an overview of the pilot that we ran and the outcomes. Some of the work that the pilot did was to directly link grant information to the data, important for us at NIH and perhaps important to you as we roll out our data management sharing policy. We assigned a Civic DOIs, digital object identifiers, and then there were many applications that were also part of the regular Figshare, but we also enabled expert guidance and metadata and improvement with customized metadata and support for large files. And so our goal here was just to understand in what way would this fill or not fill a data sharing gap. And here is the results of what we found. 21 of our institutes and centers, without any additional work from me or in my office, used the pilot. And you can see by the size of the box, the number of data that were contributed to our pilot, with NIAID being the largest contributor, but many ICs contributed data and shared data in this way. Interestingly, there was a fairly large number of NIH intramural researchers who used this generalist repository. And so that was something that we were pleased and surprised about, because the NIH intramural community is only a small subset of the entire research world. The key takeaways from our pilot was that researchers are gaining more confidence in sharing the data through repositories. And so we did find that there was a fairly reasonable amount of data sharing through our generalist repository pilot. However, in working with researchers who were using this pilot, they needed more education and guidance on how to describe data sets and the metadata fields in an effective and efficient way. And finally, when we started to do metadata enhancements that enabled greater discoverability.
Hi, Susan.
Oh no, the slides are not advancing. Oh no, what slide are you on? Are you at the very front?
We are at the optimized funding for NIH data, and then under the historic funded through research grant.
Okay, so sorry. Okay, thank you for letting me know because I'm not monitoring the chat. So you're here, correct?
Yes.
Okay, so where you need to be is here. Is this what you're seeing now? And it's going to the FOAs.
No, we are still on the same slide. Right now it's changed.
Okay, clicking through. Is it okay? Are you now seeing FOAs?
Yes, funding opportunity.
Yes. Okay. Great. Okay. Really quick recap. See, this is part of the joy of working at home. Is all kinds of things happen. So these are the FOAs. And by the way, I will absolutely give you these slides because I know that you're going to want them. You're seeing the general suppository fig share. Oops, sorry, that went fast. Are you seeing a fig share slide?
This is, yes, the pilot affair generous. Yes.
Okay, great. And now you're seeing our pilot results.
And it's the repository contains data funded by 21.
Awesome. The big boxes and the little boxes, and how ICs contributed to the pilot. Again, it was completely and utterly random. We did nothing to encourage other than to let people know it was out there. It was our way of understanding the landscape. And then you're seeing the outcomes. I hope.
Yeah, the key takeaways.
Awesome. Okay, it's working. Okay. You know, I love this slide and I love the concept, and I love the fact that you're going to have the CEDAR group come later and talk about metadata, because the concept that metadata is truly just a love letter to our future research selves, telling us as much information about the data in a coherent and computable way so that we can enhance data discovery. You know, we are working quite a bit at NIH now with COVID on creating a way to look across repositories, and it comes down to the data standards, the common data elements, the clinical research, and the metadata. And so I love the concept, and I love the fact that you're going to be thinking about enhancing metadata for greater data discovery. And there's been a lot of good work funded by BD2K in this area that we can absolutely build on. So that's data repositories, data science policy, computable metadata. Let's think about putting data on the cloud, so harnessing the power of the cloud. There are many good reasons and some difficult reasons or challenges in putting large-scale data on the cloud. Some of the opportunities is that it presents computational biomedical data science at unprecedented scales, and I'm going to show you an example of that. Partnering with industry allows us to take leveraging their cutting-edge technologies, and frankly, when it comes to AI and other things like large-scale search or BigQuery, they're really cutting edge. Plus, we're taking advantage of some of their leading-edge security tools. We can put a lot of data on the cloud, and we can also pay for that, and that creates another challenge. There's a whole community of researchers working on the cloud and in such a way that it breaks down disciplinary silos. And finally, we can build and adopt and develop new tools for cloud research. So there's a lot of great reasons, and there are some challenges that we can get into during our discussion. We have a partnership with AWS and Google through STRIDES, our science, technology, research infrastructure for discovery, experimentation, and sustainability. Kind of a lot. But what it is in three main elements, and most of this is around data storage and compute. That's mostly what STRIDES is. It's a discounting service for you to get discounts on data storage and discounts on compute. In addition, we offer training and education for researchers And as you'll see, we've trained a very large number of people to use cloud, and we'll continue to do so. And finally, we are able to partner with these two industries to employ enhanced workflow pipelines in the cloud, artificial intelligence algorithms in the cloud. So it's been a good partnership. Here's how you, as a researcher, can take advantage if you so choose to go through Strides. Oh, first I have to tell you the results. So as of this past August, we have 37 extramural institutes, like Johns Hopkins, for example, Yale, who are partnered with us in Strides. We've put almost 280 projects, programs in the cloud. We've trained over 2,400 people, everyone from undergrads to very well-established, experienced biomedical researchers and PIs. We take our training to you, or at least we used to, but now we do it all virtually. We've stored 80 petabytes of data on Google and AWS together. So it's not an equal distribution. People choose AWS or they choose Google depending on their research needs. We've used 30 million compute hours on Google and AWS. And we saved NIH approximately $9 million that results in the opportunity to spend money on different or enhanced sciences. So from our point of view, it's been a good year. We hope to continue next year with perhaps even additional cloud service providers in the Strides portfolio. If you're interested in Strides and you would like to learn more, of course, you can talk to our team at CIT, and there's an email right down there at the bottom of the page. You, as an investigator, work with your institution. Strides roles at the institutional level. So when the institution is rolled, you can work with the reseller. You have a grant. You're able to select your cloud vendor, set up an account, configure your environment, and you're good to go. And that is what we hope. We offer a number of tutorials. I hope that you will engage us in our tutorials. You do not have to be a part of Strides to be a part of the training and professional services. What have we done with this awesome power of AWS and Google? There's quite a bit. I'm going to give you one example because it's brought to our attention some of the biggest challenges and some good opportunities. So we've moved about 37 petabytes of public and controlled access sequence read archive data in Google and in AWS. So that's every little snippet of sequence from the coronavirus, SARS-CoV-2, to chickens, to microbes, to humans. It's all in there, and it's quite a bit of an interesting experiment. This is a very expensive endeavor for 37 petabytes of data, so we have had to think of creative ways of storing that data. And we now partition the data between hot active computable memory and cold deep storage. Data that is new and fresh is in hot active memory, and then it slowly migrates over to the cold glacial storage, and you can request it and we will thaw it out for you, and that this happens in a seamless way. So just working with Google and AWS to allow partitioning of data and active movement of data in a managed way has been a good partnership, but it's been something new for all of us. But this is our way of bringing down the cost of storing very large amounts of data in a commercial cloud partner. Well, this is one example of what we've done from the computing analysis of this data set. So in a partnership with AWS, Ceratus has taken all of the sequences from SARS-CoV-2 that have been sequenced across the world in 10 years, and they put it into a bucket. And now we want to do an all-against-all comparison of the SARS-CoV-2 in this time frame in these data sets. Normally, that would have taken about a year. I think it's about 3 or 4 petabytes of data. It's really quite a large data set. So with the help of AWS, we were able to redirect that all-against-all comparison into a 3 to 4 day time frame. And so everything is on GitHub. It's linked to this publication, but this is just one example, and it points to another opportunity, another grand challenge, which is computing on petabyte scale data does require some serious engineering skills, and I know this community is up to it. Searching across 34 petabytes of data is another grand challenge, and it's something that we are working on, and hopefully we will be able to address within the next year or two. And I think the community, not just at the NIH, but across the community of DOE, NSF, DARPA, are also thinking about searching across very, very large sets of data. That computable metadata and the way of organizing data is now going to be incredibly important. So I love the thought of, I hope you don't see that. I love the thought that I think it was Glenn van der Linden who said that data is the new oil, AI is the new refinery, and the cloud are the pipes. So just refactoring that just slightly, data is the new oil, then software is the new refinery, and many different types of pipes can be enabled. And so what is NIH doing to enhance data for open science and really bring data from the academic software world to something that's not quite industry scale, but is not quite academic either. So a little software engineering. I know that my friends at NSF often talk about the gulf or the barrier between academic and industry software, and we recognize that, and sustainability of software is an important point for all agencies. So this past year, we ran supplements to existing software that was funded by NIH, and all ICs participated in our Tools for Open Science. The goal of this program is to enhance software engineering, to bring new partnerships and new collaborations to biomedical researchers and the software engineering field, be they in industry or in academia, to enhance their tools and make them, if at all possible, cloud ready, or at least community, large-scale community enabled. Working with Strides was not required, but we certainly appreciated if folks were working with our cloud partners. And you can see that there was a funding announcement at the bottom. I fully anticipate this will continue in the future. So what did we fund? Across 12 different institutes and centers, we funded tools to make bioinformatic clinic software more FHIR, fast healthcare interoperable resource enabled. FHIR is the way to connect electronic healthcare records from different industries. It's now becoming more mainstream in our informatics and clinical informatics research area. Getting FHIR to be enabled for phenotypic information or case reports is still a big area of research. We enabled a number of cloud commons, including FaceBase for facial recognition. From my own field, from biomolecular simulations to force field refinement, to the study of blood flow, all of these tools were funded. Genomics and large-scale genomics, pangenomics tools, neuroimaging, and also funded from NIDDK was Dr. Sauter and his tools for histology. And I threw in one of the images from his work. So we hope to get additional applications in FY21 and fund more activities, more tools, and more researchers. And this software sustainability program sort of has a life of its own, where other ICs are now picking up additional applications. So do let your community know about this program. There is time to talk about artificial intelligence, and I think perhaps you'll be interested in some of the work that NIH is going to be doing this coming year in AI. You know, AI is, probably don't have to tell you, it's sort of omnipresent. It's everything from extracting information and text mining, for example, in electronic healthcare records, interpreting genomic sequences and looking for mutations and understanding the function of proteins from those mutations. The sweet spot of AI is in reading medical images. There was just a paper out recently about looking at osteoporosis images, MRIs from knees, from patients across, I think, 10 years, and trying to develop early detection of osteoporosis. You know, usually I guess you only know you have it if you have pain, but having early signals that you might be a candidate for osteoporosis would be very helpful. here are some areas that AI is also making impact in that you may not know. For example, monitoring sleep or vital signs and giving early warning detection to doctors and nurses, or determining from phone calls which children might warrant family support. So some prevention and for a child at risk. So these are just a few of the funded activities that NIH is currently engaged in with AI. In the COVID world, a partnership with our clinical center, NIAID and NCI, is working with NVIDIA. NVIDIA is a leader in the AI classification for images, and here they've turned their high throughput algorithms on data sets provided from Asia to detect COVID-19 viruses in patients. This is early work in the COVID experience. I think we've gone quite a bit further since then, but starting back in May when we were really quite desperate to get any type of data from COVID, this pipeline was stood up. So in response to a report from our NIH Artificial Intelligence Working Group, and I've linked out here the final report, we have proposed a new Common Fund initiative, Artificial Intelligence for Biomedical Excellence, AIable or ABLE. The entire recording is on May 15th. And if you go to about 1 hour, 25 minutes into the recording, you will see the concept clearance, which I'm going to summarize for you. The recommendations come in 3 big buckets: collect good data, have really good metadata or data sheets and model cards, and make that data reusable. Don't forget about the representation and the ethics for those data and for those algorithms, and train new people in AI and make them really cross-trained. And so this is the more detailed, creating flagship data sets, making sure that when we do start to create those data sets, that it's a partnership between biomedical researchers and artificial intelligence or machine learning researchers. So at the beginning, at the onset, create the data sets that are AI or ML enabled. Publish metadata about those data sets. And here the working group gave us some very specific examples of what they wanted to see in what they called data cards. So publish best practices about data sets and model cards from algorithms and from data. And finally, this is when you can expect to see FOAs from this Common Fund initiative. So we anticipate to get out data design centers to create those high-valued gold data sets that are AI ML enabled in the early part of FY21, so probably in December to February timeframe. New tools to enhance data sets and supplements to existing awards. So I'm looking forward to working with the community on the data design centers, on the ethics design centers, and on the training design centers. Speaking about partnerships and data science, I do want to just let you know about a partnership that we have enhanced with NSF. And this is in Smart Connected Health. And here we're accelerating our innovations in computer and information and engineering by partnering with NSF. This partnership brings new researchers to NIH in the computer information sciences. It enables cross-training, and it adds a lot of value across NIH. So 23 of our institutes and centers are participating in this partnership, which I'm thrilled. It's probably the largest number of institutes and centers at NIH that have ever participated in an NSF program. So I'm thrilled about that. And here are the areas that applications can be solicited for. As you know, if you've participated in any of our NSF joint programs, NSF collects the applications through their process. They conduct the review with NIH participating, but they are leading the review. And then after the review, we partition the applications to NSF and to NIH. And so there's no joint funding between the two organizations. We partition the applications between our two agencies. We'll be interested in getting applications in information science, including methods for data integrity, provenance, security, privacy, and reliability. We're very interested in unstructured data interpretation and new AI methods for mechanistic modeling, new ways to think about design and fabrication of multimodal sensor systems, effective ways to allow researchers to participate in their own health, and of course, our programs in human-in-the-loop computing and AI-enabled delivery and information interventions. And finally, although it is a fairly well-trodden spot for AI, medical data interpretation is of great interest to the Cancer Institute. So you should be looking for these applications to come in the December timeframe. And as soon as the notice is out, I will let all of the communities know, and there's a website that you can also look for this information. So finally, before I spend a few moments on COVID, I just want to tell you what we're doing to enable research training that's different from our typical T32 or FRRK awards, which we always support. And that is, if you happen to have undergraduate students in computer information, data science, engineering, or even in economics, and they would like to spend 10 weeks either in person or virtually at NIH. We participate in the Civic Digital Fellowship Program. We've brought a number of students to NIH over the past summer. This fall, we're bringing in a whole lot more students. They are hosted across 14 institutes and centers, and they will all be working virtually. And although it is hard, we have made it happen this summer, and we're going to continue to do so because these are fantastic opportunities, not just for the students, but for us. We learn so much from these young people. And if you are interested in doing a sabbatical at NIH, we have a National Data and Technology Advancement Program. This is kind of similar in scope to what you would do if you were doing an IP8 NSF. You come to NIH to work on high profile, very difficult problems. Like, for example, we will have a scholar architecting search across petabyte scale data. We'll have another scholar working on producing integrated cloud-based platforms for genomic research that are truly interoperable. We'll be doing knowledge theories on brain circuitry, a fun program in harnessing data science for H3 Africa, of course, medical imaging, as well as catalyzing neuroscience research. So there's a lot of great opportunities. Next year, we'll have even more programs for scholars to come and work at NIH for 1 to 2 years. And again, it's a sabbatical for an opportunity for you to give to us your knowledge and your capabilities. This is everything from our strategic plan for data science. And in 5 minutes, I'm going to tell you how we're implementing that in COVID. COVID is quite an experience for all of us. We are working to put as much data as we can into existing and new repositories so that researchers can use COVID data. What will they do with these data? Better understand transmission and infectivity, evaluating new treatments and interventions, not just for vaccines, but also for therapeutics, predicting the long-term sequelae of this disease. As one of our candidates for president noted, this is a pre-existing condition. This does not necessarily go away, and we need to understand what happens to individuals who have severe or even moderate COVID infections. Linking the social determinants of health with the COVID-19 data exposures. COVID is, although it's an equal opportunity virus, it affects communities differently. Examining the impact of child and maternal health from exposures to COVID. And finally, there's just a lot of technical issues that we need to solve to really get this data to researchers in a way that's comprehensive and not siloed. And so I'm going to tell you a little bit about what we're doing. We have received a little bit of funding this year to enhance the amount of data that we can put in clinical platforms, and we've chosen to do the following. COVID near real-time clinical data from all of us. We are piloting new enrollments across the country to expand the number of participants in all of us who are COVID positive or COVID tested. In addition, we have stood up the National COVID Cohort Collaborative, N3C as it's known, to collect clinical lab imaging data from across the CTSAs and and with funding the CTSRs for inclusion of underrepresented populations in this data set. So right now there are, I believe, 60,000 patient EHR-derived data sets in this database. And from the Biodata Catalyst, we're enhancing the red and blue coral trials to understand retrospectively the effect of COVID on patients who are in clinical trials, but then are hospitalized with COVID-19. So we are putting quite a bit of data in new and existing platforms. And in order to create an interoperable way to serve that data to researchers, we are federating across these platforms. And this has required a number of new capabilities, of which I'm going to tell you about two that we are now, we've either stood up or we're in the process of standing up. And there's many that have not been stood up, and there's active areas of research, including data discovery. There's a lot of need to discover data through really interesting, novel knowledge networks or other networks and computable metadata across platforms, because trying to aggregate and find data across 3 or more platforms is not easy. They require complicated policy and data access and data request resources. Workbenches and tools across systems does not exist. So there's just an awful lot of work to be done. But here's some of the first baby steps that we've taken. Before we started this project last year, if you wanted to get data access across different platforms, it was a bit of a kabuki experiment. You'd have to get access and authentication across different systems at different times. It was sometimes could take up to 2 years. It was quite a bit of work. So we streamlined at least a bit of this. So we have a researcher authentication authorization system. It's a central system at NIH working on the JSON token model. You log in once, you're issued a passport that you can then access across at least 4 cloud systems, 4 data platforms on the cloud. And then you'll have a visa for your data that you're able to authorize. So it's a way to just streamline authentication and authorization of researchers and the data that they can access. We are also linking data across systems by a pilot that is a hashing process. For example, a researcher may have data in many different sites, the Clinical Cohort Collaborative, a clinical study from NHLBI, and maybe they're also a participant in All of Us. Linking the data across these systems will be done by creating an encrypted hash that is then decrypted through an honest broker. And this is a partnership with industry collaborators. And this is a way that we'll be able to deduplicate patients across systems and also link data across systems. This is just the beginning. We're in our first week of this pilot, so stay tuned to hear how this goes. There's a lot of policy issues that we still need to work out, but this is the early start of what we intend to do. And with that, I want to save 10 minutes for questions. So this is more or less the vision of how we are moving forward to integrate FAIR into our biomedical data ecosystem and instantiate the way that researchers can find, access, use, and reuse data, and make it more interoperable in the platforms that house this data more interoperable. This work is not done by one person, but by many, many people. And these are just some of the fantastic colleagues that I've had the opportunity to work with this past year as we've moved forward. And with that, I'm going to stop sharing for the moment and see if there are questions.
Yeah, so for the audience, you can unmute yourself and ask question, or you can just send question through the chat function. Yeah.
I can read the first question if you like. It says from Dr. McCannon, how will the data management sharing policy deposit data enforce compliance? And then secondly, validate to avoid gappy or unannotated data sets entering the system. Okay, so enforcing compliance. We have both carrots and sticks. So of course the carrot is that you will be through data sharing, you'll be able to, of course, make the community aware of your data and have the opportunity to enable FAIR, but also when it comes to renewing your application, non-compliance could in fact be viewed negatively by your program officer. Compliance will be held currently right now with program staff. And so that is where the final enforcement will be. And how will we validate gappy, under annotated data sets? Unfortunately, that is not something that we can easily do right now. We have the opportunity to ask researchers to link in their progress reports their data sets, but sort of managing the quality of that data set is not something that NIH at this moment will be able to do. So we are going to have the issue of poor quality data and underutilized metadata. And I do recognize that as an issue.
Directors would need to come in at some point, right? But at a much lower.
I hope that answered your question, Neil.
Yes, thank you, Susan.
Absolutely, pleasure. Additional questions?
Yeah, are there additional questions? You can type in or just unmute yourself. I think I don't see any more questions. If you have any more questions or would like to discuss, please feel free to send us an email after the presentation and I can forward it to Susan. Yeah, we're glad to do that. Yeah. So thanks for Susan for the great presentation and thanks for everyone joining today's webinar. We have learned a lot about the FAIR data, data science and data management from NIH's perspective. And thank you so much. Like I just mentioned at the beginning of the presentation, a recorded version of this webinar will be available on dkNET YouTube channel. And then slides will be also available through the slide share. So, I have two announcements for our next webinar will be on Friday, October 23rd. Dr. Tudor Oprea will introduce you a useful resource called PHAROS from Illuminating Druggable Genome, IDG program. PHAROS is a web interface that integrates information about understudied proteins so that researchers can easily access the information, catalyzing your own research. PHAROS can also help researchers find new proteins that may be of interest. So don't miss it. You can find the webinar registration information at dkNET webinar page. The second announcement from dkNET is we are developing a new tool called Resource Watch. This tool allowing researchers find available issues and validation information about cell line antibodies. And will also allow research to report the issues. So we are recruiting beta testers. If you are interested in helping us test this new tool, please contact me and the email address is info@dknet.org. So we'd like to encourage you to share today's webinar with your colleagues and students. To receive upcoming webinar information, you can subscribe to our newsletter, follow us on Twitter, like us on Facebook, or check announcement at dknet.org. So I want to thank Susan again for your time and then to give us so great presentation. And thank you all again for joining today's webinar. I wish you all have a great weekend.